All right. Good afternoon. Good afternoon. Good afternoon. Mm, okay. Dallas, I'll check on that. As far as I know, the last time I checked the Nearpod, I had updated everybody that but I'll check again today. All right, and folks, I am if you need me to open something, I am opening them until Friday. And if quite a few of you have some things that you need that would be in your best interest to update. All right, yeah. So the Nearpod Dallas, last time I checked, you still needed to do that. Last time I checked, not just you, Lucas, Ethan, Isabella, Dallas, Charlie, Akari, all of y'all needed to. The plant and animal cell Nearpod, that's still empty as far as last time I checked. Now, if you did something since last week, um, let me know. But that, and that's something I put in manually. That's not something that you can submit. Um, Dallas, the only other thing I could see was the scientific method, and that still is outstanding. If there was something else, it's been upgraded. All right. So I'm looking at most of you have submitted your cell transport sheet, and I'm not going to look at that yet because I want to um, I want to make sure that you understand endocytosis. So we're going to look at that first. All right. Couple of other what they call housekeeping things. So let's look at change pages. Page. Yeah, this class is going to keep me busy because I have to call home if you're not passing. So, not a happy camper with this group right now. All right, I wanted to start here and say that fifth period exam is next Monday, just to remind you. So I'm looking at your homepage. This is a reminder of the dates of all your exams. So we are having a 9-week test. We do have to have one because you are an external exam class. So all external exam classes have to have 9-week test, and it does need to be in a format that would resemble the rigor of the end of course test. Just a heads up on that. So yours is on the 5th. We are working on cell transport. And well, I can close that and start here. Already had it pulled up, but I can pull it up again from here. Alrighty. So this is in your playlist. And
Hello, welcome to Bite Size Med. This video is on
just giving it a minute. No, all tests are done here. Even when we do the in April, when they do end of course tests, the Lighthouse Virtual Academy has a special location where you test with Lighthouse. You don't go back to your home school to do that. You test with this school. Your scores may go back to your home school, but you do test with Lighthouse. Okay, and they'll give you information as to what to do, where to report, and how to do that. All right. Okay, now actually I wanted to show something before we did that. So what we are looking at right now I guess I have to come back to that. All right, before we do that, let me do this. Speaking of the test, so what we need to do, biology does have review pages, so you can look at your review page for each module to study, but the simpler thing to do rather than to look at that. So you can look at that, or you can look at the overview page. And I think it's a little easier to look at the overview page because the test is not formatted the way the review sheet is. So the overview page gives you the main ideas of the module, and we've done 3 modules now. So you need to know what is the scientific method. We didn't do a lot of graphing and metrics, but you do need to know lab safety. We did talk about the scientific method in terms of all of these vocabulary words. So do know what that means. We did talk about the cell theory and lab safety. So again, look at what is the scientific method and what are your safety rules, and know the vocabulary. So that's unit number 1. Unit number 2 is our macromolecules. And again, you have a review page. You have a graphic organizer with all your macromolecule information on it, so study that. In addition to that, you want to make sure that you here, whoops, did it not change pages? It did not. How do I get back on lists? Okay, hold on. Same module. It just popped back onto the same one. I want macromolecules. All right, so for macromolecules, again, you have a graphic organizer that has all of this information on it. So, okay, so study that for this unit. That's what you need to know for that unit. Next module. And for those of you who didn't know, a copy of that macromolecule organizer is here in the chart, in the modules. All right, so just for that unit, just study this. Okay, study that. All right, so that's unit number 2. Unit number 3 is the one we're on now, and that's cells and cell transport. Again, look at the overview, but for this, you want to make sure that you do those 2 worksheets that you were given. So the overview lets you know that's Cells are the basic unit of structure and function. They work together to create tissues and organelles. And we're not going as far as the microscope. We're going to do the microscope after, but you do need to know all the parts of the cell, what all the parts of the cell do. And so that is that. And we are today talking about these words right here. That's what our actually starts from here. All right, so all of this is what we're talking about today. Okay, so all right, so again, there is no study guide for the test. This is the study guide, looking at the overview module for each one, looking at the worksheets that you already have. You already have pictures of the cell. You have labels. You know what the cell parts do. And so now we're going to, and we're working on this worksheet right now. So I come into this. Actually, it's easier for me. This is a long module, so. So have that worksheet handy. Even if you've already turned it in, have it handy. Cell transport. Like I said, I do see a whole column where everybody, except a handful of people, and for some of y'all, I might end up pulling out into a separate session, because some of y'all are not doing any work at all. All right, so we did the top part together. We should have two checks in each column for the appropriate definition of isotonic, hypotonic, and hypertonic. I asked you to draw pictures of the cell, and actually those pictures are also in the module. So you are supposed to go back and answer these questions. In this section, there were 4 words that you had to associate with each of these 6 pictures, which means some of the words are going to be used more than once. You need to remember that a gas can diffuse through the cell membrane. Water can diffuse through the cell membrane. Glucose is a sugar. It is slightly bigger. So this is going to be part of the discussion today, and large molecules of protein. Water can diffuse through a membrane. So your question is it active or passive transport? And then the same thing with these questions. You're going to want, these are multiple choice. Basically, you can just highlight or circle the correct answer with those questions. All right, so I'm going to stop there and we're going to watch the video. All right, let me come back to, which I had already set up, and now it's not.
Hello, welcome to Bite Size Med. This video is on endocytosis and exocytosis. The cell membrane is a phospholipid bilayer. Stuff moves across the membrane to either get into the cell or to get out of it. Small things like ions, they can use protein channels and they can diffuse. But what about macromolecules?
All right. This is also part of the review. This topic of transport is on next week's session. You can consider this part of the review. We've already talked about what a cell membrane is, the vocabulary, bilipid layer, which lipids are fat molecules. So each one of these little blue things with the tails, those are a lipid monomer. And bilayer means there's 2 layers of them. So the cell membrane is made up of a bilipid layer, 2 layers of lipids. Okay, vocabulary, review.
They need another way, and that's by using vesicles. These vesicles pinch off the membrane. If they're going to bring things into the cell, the process is endocytosis, because endo is internal. And if instead the cell is throwing something out, that's by exocytosis. Exo is external. And these are the methods of bulk transport. There are different kinds of endocytosis. It could be phagocytosis, pinocytosis, or receptor-mediated endocytosis.
All right, I'm pausing again. So what we did yesterday or Monday for you all was passive transport. Now we're on the other side talking about active transport. Remember, passive transport didn't require any energy at all. And it also included facilitated transport, ways to get things into the cell without much energy. But what we have now is sometimes there are objects outside the cell that the cell needs, but they're too big to just flow through the cell. So we need these methods of endocytosis to try to get them inside. The other thing that you have to remember is the rule about concentration gradient. Things go from high to low. What they're going to talk about now is the fact that if it's outside, sometimes it's in low and it's not, and inside is high, and that's against the concentration gradient. So they are being brought in, and to do that, you need energy. So active transport requires energy.
Phagocytosis means cell eating. This happens only in a few kinds of cells, like in the immune system, neutrophils and macrophages. What do they eat? Pathogens like bacteria, tissue debris, etc. Whatever the substance may be, when the cell comes in contact with it and it wants to engulf it, it will form these extensions called pseudopods. They go round the substance, meet, and fuse. Now it gets pulled into the cell and separates from the membrane. Now this cavity, lined by that phospholipid bilayer, is called a phagosome, and it's got the engulfed material inside. That needs to be broken down. Among the organelles, which one does that? The lysosome. The phagosome fuses with the lysosome to form a phagolysosome. These lysosomes have hydrolases. They will digest the material, and what remains that's undigested, that cannot be broken down, that forms the residual body, which will then get extruded from the cell. This extrusion is by exocytosis, because the cell is throwing its contents out.
All right, I want to stop it there. All right, so vocabulary words, pseudopods. Pseudo means false, and pods usually with cells, it means feet or feet or hands in this case. All right, so what they're saying is the cell membrane actually forms these extensions around whatever it wants to bring inside. So it creates a pocket for that thing to come in. And then eventually the pocket closes and it pinches off and forms that phagosome. Okay, so it forms a vesicle. And then the vesicle, once inside the vesicle, things will digest it or do what it needs to do with it. So that's in. So again, into is endo. Think of that prefix endo as into, and think of the prefix exo as exit. So things on the other side here now, they're spitting things out that they don't want.
So that is phagocytosis. Pinocytosis is similar, but it's different. Instead of eating, the cell is going to drink now. This is fluid phase endocytosis. It gulps in the extracellular fluid with its dissolved solutes. So the vesicles here are smaller. These are called pinocytic vesicles, or pinosomes. They will then fuse with an endosome for further metabolism. Unlike phagocytosis, pinocytosis is seen in most cells, for example in the small intestine. But it's very non-specific. The cell is just gulping down the ECF with whatever is in it. This process can be more refined with receptors, and that's what happens in receptor-mediated endocytosis. Now, most forms of endocytosis use something called clathrin to form the vesicles. This clathrin has a triskelion structure, so it has 3 legs. It's a 3-dimensional structure, so it forms a cage around the vesicle. Receptor-mediated endocytosis usually involves clathrin, and if it does, it's called clathrin-dependent endocytosis. The cell membrane has depressions. Clathrin is located beneath these pits. These are coated pits. They are coated with specific receptors. The two are attached by things called adaptins. So when the ligands bind to the receptors, the vesicle is going to form. It's more specific than fluid phase endocytosis because a specific receptor is involved. Now, as the vesicle forms, there's another molecule called dynamin. That's located at the neck of this vesicle. It's a GTPase, so it uses GTP and helps pinch the vesicle off the membrane. This vesicle then loses its clathrin coat. So that's uncoating, which is done by an uncoating ATPase. So it uses ATP to remove the coat, and the clathrin can be used to form another vesicle. After uncoating, it fuses with an endosome to form an early endosome. The endosome has proton pumps on its membrane. They send hydrogen ions into its lumen, keeping the environment acidic. That acidity helps the ligand detach from its receptor. So it's like a sorting center. The receptors are going to get recycled back to the plasma membrane. What's left behind are the ligands in the endosome, which is now going to form the late endosome. That late endosome will fuse with the lysosome. The lysosomal hydrolases break down and digest the content. A classic example used for receptor-mediated endocytosis is with low-density lipoprotein receptors, which take up LDL cholesterol from blood. It goes through this process in the cell and gets broken down to cholesterol. So that's just an example of receptor-mediated endocytosis. Clathrin is the most studied in endocytosis, but there are clathrin-independent methods as well. For example, caveolae in endothelial cells. So there's lots of different kinds of endocytosis, but all of it would involve things entering into the cell. What about leaving the cell? What would the cell want to throw out other than waste material? Proteins, hormones, neurotransmitters, things like that. Stuff that it synthesizes and has to leave the cell and go somewhere else and perform its function. Proteins get synthesized in the cell, and then they go through all modifications through the endoplasmic reticulum and then through the Golgi apparatus to get packaged into vesicles. If the content of these vesicles has to leave the cell, it will do so by exocytosis. There are different pathways for that, like there's the constitutive pathway, where when it's synthesized, the content gets released. There's no storage. These are called secretory vesicles. The second type is regulated, where the content can be stored, such that when there is a signal, it can get released into the extracellular space. An example of this is at the synapse, like the neuromuscular junction. The neurotransmitter is stored in vesicles, and when the action potential arrives, calcium enters the cell and makes the vesicles fuse with the membrane. The fusion of the vesicle to the membrane involves different proteins. One complex is the V-snare T-snare complex. V is for vesicular and T is for the target organelle. Along with this, there are other proteins and GTPases. Now this complex, it helps with docking of the vesicle onto the membrane so that the two can fuse and the contents of the vesicle can be released. There are actually different ways that this could happen. One would be where the vesicle briefly opens, releases what's inside, and then closes. So it can be recycled and used again, which is one of the ways that exocytosis happens at a synapse. Another way would be complete fusion with the target membrane, and then releasing the contents of the vesicle. If it does fully fuse, in exocytosis the cell gains membrane, and in endocytosis a bit of the membrane gets back into the cell. The two thus couple, so the cell size stays the same. And that is endocytosis and exocytosis. If this video helps you, give it a thumbs up and subscribe to my channel. Thanks for watching. I'll see you in the next one.
Okay, so this is in your playlist. Alrighty. So what I need you to do, if you haven't already done so, and that's a couple of you, is to make sure that you have completed this worksheet. So I specifically wanted to say that that video went with this down here. So the three topics that were mentioned there, the words endocytosis, exocytosis, and pump. Those are the three words that should go down here on this level. And so if you didn't do that, then you need to go look at your worksheet again and resubmit. So again, this one is saying, is they're eating, taking stuff in. This one, the picture is showing one, two, step one, this vessel is inside. Step two, it went to the side and spit it out. And step three, the cell membrane is going back to normal. The last picture, it is showing that it's going from low to high and has to be pumped in. So the line in the middle is your membrane, and all those molecules are, remember, when you go across against the concentration gradient, you're going from low to high. It's low on the left side, high on the right side, and normally for diffusion, it goes from high to low. And when it goes from high to low, it flows without energy. These processes at the bottom, they require energy. Here again, they're showing you the cell membrane, and notice the arrow again is showing you that the molecules on the outside are trying to pass through this little transport protein to get on the inside. So what are we calling this? This diagram up here is looking at just water. So what is the movement of water called? And again, it's going from low to and look at the way the arrows tell you which way the things are moving. The one above that as well. You've got arrows. You needed to explain, put the words hypo, hyper, and isotonic underneath each picture in case you had not done that. And again, these over here are fill in the blanks, but they give you choices. Okay. So if you have not completed this worksheet, it's due today. So please, like I said, most of you looks like you have turned it in, but make sure, look at yours again to make sure that it says what it's supposed to say. Last chance to change your mind. After this point, I graded it as is. Okay, so make sure you did that correctly. You match these terms correctly. This is vocabulary, so this is your study sheet for this topic. All righty. Any questions? Any questions? Right. If you are finished with that and you feel like what your worksheet was already is as good as it's going to get, then go back and use this time to review the modules. Like I told you, review, make sure you look at your cell uh, worksheets. Some of you didn't fill them out properly, so you need to do that. And your macromolecules. Go back and review your cell theory. What is the cell theory? What are the lab safety rules? You had a picture that you were looking at all the safety rules for lab safety. What was that? Scientific method. Make sure you understand what scientific method is. So use this time to review. You got another 15 minutes. So if you've completed the worksheet, review. Your test is on Monday. Friday at 1:30. If you need to talk with me, that's when I have tutorial time. So you don't have to sign up. You just go in. I'm present. Just log into the Zoom. I'll be there. Some of you might extend an invitation. I'm looking at this grade book and it's not making me feel warm and fuzzy. Desianne, I'm just seeing this message. Did we meet on Friday? No?
Did you call my name?
Yes, I'm looking at this. It's that you were asking for a tutorial meeting. Did we meet?
Huh?
Did we meet or do we need to meet?
Oh, we already met.
Okay, good. Look, I'm just trying to make sure. Okay.
Oh, okay.
All righty. Where's Charlie? Charlie is absent. All right, Ethan, have you caught up on all your work? When you're in this class, please do the work for this class, please. I have opened up several of these assignments so there, if you have missing work that needs to turn in, the assignment window is open. Still late. Some of these are so late, it's going to give you 0 anyway, but I'll excuse it and give you half credit, but that's better than a 0. Because after next Friday, all of these zeros stay zeros and we start a new term. So we're not looking at this semester again. Moving on. Okay. Dallas, which quiz is that? Um, yeah, I could give you, that's the one that needs a code. Quiz, macromolecule quiz. The macromolecule quiz. Okay. 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 Gotcha. Let me open that. Do that right now. Oh, if it's not in there, you didn't do it. Let me. All right, Dallas. Hold on. I have to change the date. Open until the 20. Actually, it's open until today, so I really don't need to change. I'll change it till tomorrow. Save. And I just need to send you the code. Cell transport, I haven't checked yet, and I was waiting to give you all a chance to make sure that you had the information that we discussed today. So I will check those today. And for most of you, that should be the only assignment that I have to check. In the meantime, if you know you've sent it, don't worry about that one. Okay. Okay, Dallas. All right, Dallas, there's a code for that. And like I said, that is open until tomorrow. Let me see if there's anybody else who needs to do that. That person. Brandon, you also did not do the plant and animal quiz. And Ethan. So you have until tomorrow to do that, and I'm putting the code. So I need you to do that right now, because that's going to close tomorrow, and that will remain as 0. All right, there's the code to get in. So right now it should be. Did you take a second attempt on that? Because you must have guessed your way through it. Yeah, you did. Okay, let me check this. Why? All right. Okay, Ethan, I got you. For some reason, yes, it's in Canvas. You should. Okay, give me 1 second. It should say, what's it called? It's called Plant and Animal Organelle Quiz. So yes, it's in Q1, 2, 3. All right, Ethan, I got yours. It's a good thing I checked. All right, who else did I? Oh, is it time to go already? Because I got somebody from the next. Amelia, log out and log back in. I'm still in my 5th period class. I just realized you're in sixth period. All right, let me see who else I need to. grapple with. Okay. Before I let y'all go. Before I let you go. All right, so I took care of that one. But Brandon and okay, so it's just Brandon and Dallas. All right, so Brandon, take that code, do that test. Otherwise, that's going to stay at 0. All right, and that being said, I'll see you all tomorrow. It's time to go. Time to go.